Uh, okay. Hopefully, I'm not confusing too much. But I know that was a lot of details there. We kind of really, like I told you, we were going to kind of start heavy, and I was intentionally doing that. But we're going to keep. Yeah. Also, do you have something?
We are recording this, so it will be on the Kansas Soil Health Alliance YouTube. So I know a lot of guys already asked us about that. It will be available later.
Um, so yes, yeah, I'm intentionally putting all this up front. So if you're feeling a bit overloaded already, don't stress. We're going to keep circling this around. I'm going to keep filling in the blanks in the bigger picture. It'll start to sink in as we go through the day, I'm sure. So, okay, let's now bring in a practical element to this in terms of how we can manage some of all of this nitrogen metabolism stuff. How do we put some of these ideas into practice? And one of my most favorite strategies for this is a foliar spray, foliar applications, for some reasons we'll kind of discuss now. So why there's a lot of kind of benefits to this, or a lot of opportunities here. Yes, you can improve nitrogen use efficiencies. When we talk about nitrogen loss, be that leaching of nitrates, be that off-gassing of ammonia or those nitrous oxides, this primarily happens from the soil. It is the soil where most nitrogen losses happen. So if we can do a foliar application and direct that straight into the leaf, straight into the plant, you're keeping at least some of your nitrogen. And I'm not saying we're going to do 100% foliar feeding. You're always going to have some. Like I say, I think your starter applications, the early applications, it's best through the soil. Get the plant established, get that canopy. Once you have a canopy, then the opportunity is to finish that crop with foliars, with really high efficiency, low input foliars through that pathway once you have that canopy. But I do acknowledge we need some nitrogen now through the soil to get things up, to get things going. So I'm totally fine with that. But it is a double-edged sword. The nitrogen that we put into the soil is prone to be lost. The nitrogen use efficiencies, they typically hover around about 50%. So about 50% of what you apply enters the plant. The other 50% is not all lost. Some of that will be held and stored in the soil organic matter, or stored in the bodies of the microbes. So some will be stored, but some will be lost. And that's what that's the opportunity with foliars to say, well, if we direct this straight into the leaf, we are keeping some of that nitrogen out of the soil and therefore minimizing some of those losses. When you minimize losses, that means you're increasing your efficiency. So you can start to use less. And that's the economic gain that then comes with that foliar. So yes, you can improve yield, and we'll share some examples of this, or quality, or that metabolism that we were talking about. Reduce those environmental losses. This is good for our waterways. As you all know, we have too much nitrates in the waters, in the groundwaters, in the rivers, etc. We have too much of that, too much nitrogen in the air, etc. But also with foliars, we can improve that nutritional density of the foods that we grow for consumer or feed for the livestock, improving the mineral content there through direct applications, improving the quality from a nutritional point of view of the food and feed. So, and the point is that when nutrients are in the soil, it doesn't, just because they're in the soil, doesn't guarantee that they're coming up into the plant. Nutrients have all sorts of interactions down there in the soil. There are problems with uptake. Those nutrients can be locked up. pH extremes, reducing the availability of those. They can be bound. There are various nutrient antagonisms, nutrients competing for uptake. So there are all sorts of those kind of interactions, maybe poor soil temperatures, poor biological activity or cycling, these types of things that can then limit the availability and the uptake. So just because the nutrients are there doesn't mean that they are being taken up. Poor soil biological activity, or if the plant is a bit stressed, all of these things can kind of limit uptake. And here's some examples of the nutrient use efficiencies of some different nutrients. And you can see here nitrogen. They say here on this kind of study here, 30 to 50%. I think 30 is probably a bit harsh. I think it really hovers a bit more around that 50%. But phosphorus even lower, 10, 15, 20% of phosphorus inputs get into the plant. Now, the rest is not so much lost like nitrogen can be. This is the reason nitrogen is particularly a hot topic, because it is so easily lost. It's such a leaky nutrient. Phosphorus mainly just locks up, and it doesn't really leach very much. It doesn't off-gas. So it's the only really way we lose phosphorus is through soil erosion, physical removal of the soil. So you're not losing it. At least it's there. It's locked up. You can unlock it in time. And you can do that through better soil biology. And that's your soil health principles, that's your soil biology discussions, all of that kind of stuff that we could then go and dive into. Yes, you can use biology covers and biology to kind of unlock and leverage some of that fertility that you already have in the soil. So phosphorus, also very low efficiencies. And same with these others, potassium, it's 50 to 60%, sulfur 8 to 12, and then we hit the trace elements. And this is kind of the big ones here. It really drops down to a couple of percent. And that's really mainly, trace elements are very prone to locking up, and again, particularly in high pH soils, which I hear a lot of you guys are a bit more on that spectrum here. And so those trace elements are very prone to these lockups, but also it's just a sheer factor of that they are required in such small amounts. So when we put such small amounts of nutrients into the soil, those small amounts of nutrients are so diluted, they're very easily locked up before the roots can kind of get them or access them. So this is why the traces have very, very low efficiencies, and this is why and I'm sure many of you probably have all sorts of mixed feelings about foliar sprays and how effective they are. And some are very skeptical of foliars. They can be a bit hit and miss, and I think that's fine. We're going to talk about why that is. But I think that even amongst the most skeptics of foliars, most agree that at least trace elements are most effectively applied through the foliar pathway, because you keep them out of the soil in these lockups and you just direct those directly into the plant. And the foliar spray can deliver meaningful amounts, because the trace elements are required in such small amounts that we can actually deliver meaningful amounts through the foliar for production. So I think those are the ones that are particularly relevant. But we're going to kind of extend beyond that and talk about some of the others, and particularly a bit of a nitrogen focus. So that's really the key. We get the efficiency gain here because we bypass some of these soil imbalances, these lockups, these interactions. We can bypass all of that and put it straight into the soil. Straight into the plant. So none of these nutrient antagonisms. We don't see these losses as the off-gassing, the leaching, etc. And ultimately this means less losses, means you're saving, so we can use less fertilizer and therefore get that efficiency gain. That's the idea. Now, I'm not, like I said earlier, I'm not suggesting that we're going to go and apply 100% of the crop and plant requirements through a foliar pathway, especially things like nitrogen. And that seems to be the big critique. Oh, this foliar nitrogen thing, you can't supply enough of the plant needs. No one's suggesting that we're going to do 100% of the plant needs through a foliar. It's about using the best all tools available. Integrated nutrient management. Get the plants established in the soil, but then be focused on those really high efficiency kind of foliars thereafter. It's a combination of both. Foliars are not a silver bullet. They're just a useful tool. Let's kind of focus on getting the most out of them in terms of what they can do. And that's kind of then how I view them. Okay, so what about in nature? In natural systems, do plants even take up nutrients through the foliage at all? Is this a natural thing? If we try and kind of have those discussions about biomimicry, following in nature's image, well, actually, yes, there are many examples of plants taking up nutrients through the foliage. The first one, obvious, sea plants, aquatic plants, kelp in the ocean, which of course is minerally rich. This is why kelp is one of the fastest growing plants on planet Earth. It can produce incredible photosynthetic rates, incredible biomass production, because it's soaking in all of these mineral catalysts that catalyze that photosynthetic process and drive that biomass production through photosynthesis. So yes, aquatic plants, ocean plants particularly, are absorbing nutrients through their leaf. This one might be quite relevant in this part of the world. Dust storms. There's a really good study out of Israel, and I'm not going to go into I'm not sharing these slides today, but a really good study that showed they applied dust to wheat, to chickpeas, garbanzos, and maize. And maize had a less of ability here because it's not as an arid evolved plant, unlike the other two, which come from those kind of more the breadbasket and those hotter kind of climates. But they demonstrated that particularly wheat and chickpeas and maize has this ability too, but not as good, that the plant, when that dust is applied, the plant will actually excrete leaf exudates, acids, onto the leaf, organic acids, and it will solubilize nutrients out of the dust and take them up. And they showed in the study that the chickpeas and wheat especially will absorb meaningful amounts of phosphorus, particularly phosphorus, which had significant impacts on growth, on yield. So even dust as it deposits on the leaf, the plant can acidify the leaf surface and strip nutrients out of that dust and take them in through the leaf. So maybe that dust bowl of you guys was maybe not all bad. Hey? Make sure you catch some of those neighbors' dust. If those guys are letting their dust go, man, this is why we've got to have green cover. This is the living root soil health principle, hey, 24/7. Let's keep green cover. Let's keep not just root cover, let's keep leaf cover. We can not only capture sunlight, energy, photosynthesis, maybe catch some of that dust from the neighbor's bare field as well. Okay, so dust storms. Then we also have urine from your livestock, of course, but also even insects. A really great study showed that from insect feces that the leaves were absorbing amino acids, the organic forms of the nitrogen from the feces were being absorbed through the leaf from insects. So again, this is that broader discussion of trying to encourage as much beneficial biology, beneficial insects into the farming system, using companions or flowering plants or flowering strips or leaf margins, pollinator strips, these types of things, encouraging more beneficial insects into the farming system to provide those ecosystem services of pest regulation, predators to kind of eat our pests, but the more beneficial insects we can bring in, they can also deposit their waste products. So plants do take up those through the leaf. And lastly, even fog. A great study out of California showed that in fog there's a lot of ammonia gas. The same ammonia that you use for your fertilizer, and that they demonstrated that redwoods absorb ammonia gas from the fog through the leaf as well. So yes, there are some examples, just for kind of fun, for curiosity here. Yes, there are examples of plants in nature taking up nutrients through the leaf, and that's what we're trying to obviously mimic through our production-focused agriculture. So how do plants take up nutrients? These are the different ways in which nutrients are absorbed. I'm mainly going to talk about the top three here. So the waxy layer, the cuticle, there are these kind of micropores in that waxy layer, which nutrients can pass through. The stomata, particularly when the stomata are open, then we get more nutrients passed through the stomata. So the stomata are a key pathway for uptake. And then trichomes, these are fine hairs on the plant's leaves. It turns out this is a bit more of a newer discovery that actually the trichomes are hotspots for nutrient absorption, especially in sunflowers. I'm going to share a great example with this. And then we have a couple of others, like so the veins in the leaves. They've shown that the cells right adjacent to the veins are very permeable, and nutrients can kind of pass through there. Other leaf structures that can also play a role, or something called endocytosis. Some of you might have heard of this one before. It's where the plant can engulf kind of quite large macromolecules and things, both through the root or the leaf, whereby the leaf basically internalizes, folds in on itself, and anything that's kind of sitting on the leaf, be that a microorganism or nutrients, large complex things, it kind of folds in on itself, internalizes and reheals that, and then those things that were on the outside of the leaf become inside and where they can be used. So this is another one. I mainly want to talk about the top three, which are the perhaps more important main drivers. So, and we're going to talk about the weather conditions. Humidity is a big factor here. So in the cuticle, that waxy layer, there are these kind of micropores, and when the humidity is low, dry air, those pores are more closed and retracted. And so it's harder for the solution to get through the leaf. So when we have nice humid air, the pores are more open, so that solution can kind of pass through. Similarly with our stomata, the open stomata, nutrients travel along the cuticle of the guard cells, so they move along through the guard cells. This is an important pathway for uptake. And again, when the stomata are open under high humid conditions, we will see better nutrient uptake. Now, the stomata can still take up nutrients when they're closed. I could use the analogy of closing our eyes, closing my eyelid. Yes, if you hit some foliar spray on my eyelid here, yes, the nutrients can still be absorbed through my eyelid into my eye. So yes, closed stomata can still absorb, but when they're open, of course, that water's really getting in there. So same analogy. So you want high humidity. We're going to come back to that. But these are your kind of pathways through the cuticle and through the stomata. And then the other one here is this trichomes. And this is kind of a newer finding. They're kind of doing a lot of work to see which plants this is kind of more relevant for. and sunflowers is the kind of the big one that they've really demonstrated this particularly. So these fine hairs, these trichomes, these are the fine hairs that do all sorts of things for the plant. Some of them, they're called glandular or non-glandular. The glandular are the ones that release the smells and scents, those aromatic compounds we were talking about earlier. So some of them are designed for that. But some of them, these hairy ones, they're designed for capturing rainwater or capturing foliar spray nutrients or capturing dust and capturing those particles and funneling them down to the base of the hair. So that's really what they're kind of designed for, to catch kind of water, nutrients, dust, and bring it down to the leaf surface. And what they've shown is that you can see this nutrient droplet here. It's kind of bounced off that. It caught that and fell to the ground, fell to the leaf surface, sorry. And you can see here's the base of the trichome. Here's this nutrient droplet here. That's really interesting. What they found is that these cells right here, they're called the basal cells, at the base of the trichome. These cells specifically here, these basal cells, have very high permeability. Particularly, even just one cell over, just the one cell next door, it drops off hugely. It's specifically these cells right at the base of the trichome that are very, very permeable, and they will absorb that. And here's a nice example of this. This is from a study where they're using some pretty high-tech gadgetry to stuff I'll admit I don't fully understand, lasers and hyperspectral cameras, and bringing this stuff together to kind of visualize the nutrients in the plant, in the leaf. There's some really cool things out there happening in this space now. And so this was again on that sunflowers. So you can see here, this is a leaf transect, and we're looking at zinc absorption in this example. And the cold, dark colors, that's low concentration of zinc, and the warmer colors, white, is very high. And here we have a transect of a leaf where they applied some zinc, and then we're looking at over time. So 15 minutes, 30 minutes, 1 hour, 3 hours, 6 hours. And what you can see is around about this hour mark, we can start to see just some hot spots. There's these spots of very yellow-white that you can see here. And this is the zinc getting very high concentrated right there in those zones. And you can see they start to become very pronounced here. Well, it turns out these are the trichomes, and these are those basal cells that are absorbing that zinc. And once the zinc gets in through these trichomes, well, then you can see after about the 3 hour mark, okay, now we're getting more of the red coming through. That zinc is now moving throughout the rest of the leaf. But you can still see the hot spots of absorption. These are the key. We've determined here that zinc particularly is absorbed right there through those trichomes, and then it is redistributed. And this picture here demonstrates this beautifully. Right here we have a bird's eye view. We're looking down on one of those trichomes. So the circle here in the middle, that's the hair. That's the trichome that would be sticking up this way, bird's eye view, and you can see those basal cells that I was talking about around. Look at those basal cells right next door. Look at all these other cells just literally cells away. Low concentration of zinc, but particularly these basal cells around that hair, around that trichome, are the hotspots of nutrient absorption. So there's some cool things happening out there with visualization of plant nutrients and things. You can do this for all sorts of different nutrients. There's some really whiz-bang technology coming out there. So I think that tools like this will make the discussion about foliar sprays really interesting. I think it's really going to help us cut through some of the variability that we see with foliars, some of those challenges, as we can really understand how best to optimize. And I mean, this also opens up an opportunity for breeding strategies. If clearly these trichomes are so important for sunflowers, we could breed sunflower varieties that have a high expression of trichomes. And then we could gear and design a foliar, low input, high efficiency system around that variety that has a high tendency to suck up nutrients through the leaf. So there'd be, some interesting kind of opportunities, I think, that could come from that too. Okay, so now let's dive into the details on the mixing up of a foliar spray, how to design and how to implement a good, effective foliar spray. And I think that many farmers probably have mixed feelings about foliar sprays. They've seen variable results. I think they have that reputation. And I think that the key message here is that it's not just about saying, okay, let's get some fertilizer, let's put it in water, let's dissolve it, put it in the tank, and let's go. All right, there is a lot of specifics that you have to design for, intentionally design and manage for, a lot of variables that influence the success. We want to avoid that kind of spray and pray approach. We want to actually design for success. And that's what we've got to put specifics into these variables that influence the success. And so I break it down into these 4 things. I use an acronym called FACE: formulation, application, crop or the species, and the environment. So FACE. And when you're mixing up a foliar spray, you want to think about these 4 things, and I'm going to break down under each one. How many of these? I want to tick as many boxes as possible. I want to make sure I get as many variables in the formulation design spot on. I want to think about the application, the nozzles, etc. I want to think about who I am applying it onto and what are the current environmental conditions. And if you can line up as many of these variables, then you're going to be inching closer towards that designing for success rather than more haphazard kind of approach. Okay, so the formulation. Let's dive into this. Water quality, for sure. It's absolutely a really, really important and overlooked piece of this puzzle. If you're using bore water, groundwater, if you've got a lot of salts or carbonates and bicarbonates, if it's quite alkaline water, high pH water, I don't know if some of your water sources are. I'm seeing a nod here. If you've got those kind of high pH waters, this is not ideal for a foliar spray of anything. This will impact the effectiveness of your pesticides. This will impact you also and your nutrients. So those contaminants that are in there, particularly the carbonates and bicarbonates, they are particularly guilty here. They will bind to things when you put them in that water. So they will be binding to your chemistry. They will be binding to the nutrients and actually reducing their effectiveness, reducing their availability and therefore their effectiveness. So you want as cleaner water as possible. Now, that could be harvesting some rainwater if you have such an opportunity to collect some, maybe at least dilute some of your bore water waters with some rainwater. So you can at least dilute those salts, so as much rainwater as possible, or otherwise look at investing in an RO system. Now, the RO system I know that can be pretty costly, but it is worth it in my opinion. There was a farmer just up in Washington State there who I know who was also up there. He has invested in one. It's not cheap for his scale. It cost him about 70 or 80,000 to invest in that system. But the savings in herbicides alone, because that clean water will improve the effectiveness of all of your chemistry. And he told me that the savings in herbicide alone, in terms of improving efficiency of herbicide, he paid that off in 12 months, in one season. So one season of herbicide savings. And when I saw him earlier this week, he told me there was about 5 other guys in the room who had all done the same, invested in the RO, and they'd all paid it off within 1 to 2 years. I mean, that's a short ROI. I mean, there's not too many things you're paying off in a couple of years. And so I think this water quality is a huge oversight on many people's parts. I think a lot of our issues of herbicide resistance and all of that discussion, I think a lot of that is probably to do with poor water quality, tying up the chemical, making it less effective, leading to development of resistance. So I think water quality is a big one. As cleaner water as you can source or utilize, it is key. It really is key. And I think on the RO, I would suggest if you want to look into that, if you want to do a trial, do a test and just ask a manufacturer, say, look, I'm thinking about buying. Can you give me some free water? Maybe you'll pay for the freight. Just pay for some freight. Get some free water and a tank's worth, whatever, 3,000 gallons or something worth, and do a spray and compare it to your water and just do a small trial. See for yourself. If you see the response, if it's worth it, then you can think about investing further from there in the full kit. But it is something that I would highly recommend looking into. Okay, then we move on to the rest of the things we put in, the nutrients, etc. And you've got to think about what form of nutrients. Are they soluble? What's the molecular size? Generally, the smaller the better. Larger things are a bit slower to come in. Smaller things are a bit faster. Yeah. Oh, sorry. RO, reverse osmosis. Sorry. Yeah. And yeah, please interrupt me if there's something like that. No worries at all. Yeah, reverse osmosis water. Sorry. No, yeah, that's not some weird Australian word or something. RO water. Something weird. Okay, reverse osmosis. Yeah. Now, if you dive into a lot of the literature about foliars, and that the nutrients have to be soluble to be available. This is also, again, the classic kind of view, and it is true. The more soluble the nutrients, yes, the quicker they will pass through the leaf. So it's a perfectly fine comment to have there. But as we talked about back here, this is not soluble. These nutrients are not in soluble form. So even tiny things like dust, or let's use an example of some of those micronized products, things like ground down liquid limes or liquid gypsums, liquid phosphate type things, rock phosphate, these types of things that are technically not soluble. They're just micronized, ground down to a tiny particle size, and then mixed with water, so it's a suspension, a solution, but it's not actually soluble. I think those are still relevant. I think the plant will acidify the leaf surface, and it can still get nutrients out of those kind of liquid limes or liquid gypsum, those types of things. But generally, sure, the more soluble the nutrient, the better, or the faster. I think that leaf solubilization process, you're talking a couple of days for that to get into the plant. It might be 2, 3, 4 days, something like that. Whereas the fully soluble forms, they are going to pass through really quickly within a few hours, which we're going to talk about shortly. Now, then we've got the charge of your nutrients. All of our nutrients are positive or negative, cations or anions. And both of those are not ideal for passing through the leaf. And that's because the plant's leaf surface is negatively charged, just like your soil colloids. And so that negatively charged leaf surface means that when you apply a negatively charged nutrient, an anion, there is a repulsion there. They kind of press away from each other. Now, it's not to say that cations or anions can't pass. They do pass through, but they're not so fast. They're not so efficient because there's this repulsion of the negative to negative. When you apply a cation, a positive, there's this strong attraction and you get almost this bottleneck. And so that can also inhibit absorption. So a cation or an anion are not ideal. The ideal is a charge-neutral molecule. And I'm jumping ahead a little bit here, but this is why we then add carbon sources, or you've come across chelated nutrients, this term, chelated nutrients. That's what the chelating agent does. It binds to the cation, or the carbon will bind to those anions, and it will help to neutralize that charge. And when we have charge neutral molecule, now this will pass through the leaf most quickly, the fastest. And that's what we kind of want. So that's where we're going to expand more on this one. pH. This is also my point about your water. If you do the reverse RO water, reverse osmosis water, this becomes a bit less issue because you're cleaning up taking out all of those contaminants. But if not, I would say you absolutely need to focus on your pH here. Maximum of 6. Pretty much all of your nutrients, they will absorb through the leaf more effectively under acid conditions. And really the ideal pH, I would say, is really more around 5 to 5.5. If you can hit this zone, this is good. Maximum of 6. You want acidic conditions for that foliar spray. And this is also true for most of our chemistry. Some of pesticides like alkaline conditions, but most of them are on the acidic spectrum. And if you acidify also your spray tank for most of your chemistry, you'll also find it more effective in terms of absorbing into the pest or absorbing into the weed. Same deal. So I think the pH thing is really, really important, especially if you don't do the water quality issue.
I understand RO is probably preferable, but do you have some recommendations on like soil conditioner or water conditioners or acidifiers, or you can't do RO?
Yeah. Yes.
Would you at least do some of that?
Absolutely. Yeah. So if I can't, and so then my next would be, can you somehow invest in some water storage for rainwater collection so you can have at least some dilution? So
rain first,
yeah, well, yeah, there's that too. So then beyond that, yes, it becomes very important for the water conditioners. So of course, I'm going to circle back to this, but we often use AMS for this to help bring that pH down. I think that's good. AMS is a good option here, but I would particularly use citric acid, or maybe a combination of AMS and citric. And the reason that I say citric is an organic acid, which means it is a carbon-based acid. So you're getting free embedded carbon with that acid compared to ammonium sulfate where there's no carbon. But citric acid is a good acid for this. It's a very soft acid. It's great for biology. It's a common root exudate. It actually feeds the biology. So citric acid I would be very much leaning towards, or a combination of AMS and citric. And yes, I would add these first. put them in first to bind to all those contaminants and salts. You want to put them in first to do all of that, and then all of your nutrients and carbon and things get added thereafter.
I guess you would like just pH strips to test your water to try to get it in the 5 to 6 range.
Correct. Yes. Yeah. Yeah. You can get one of those little handheld battery meters. They're not too expensive, a pH meter either. So yes, correct.
Would that be adequate also for
Yes. Absolutely, absolutely, yeah, and that's true for your pesticide applications too. Get those acidified done first, get those things bound up, and then add your chemistry and all your nutrients. Yeah, for sure. Okay, the concentration of your nutrients is of course key. If it's a bit too hot, it can burn them. If it's too dilute, maybe not so effective. So this is kind of an EC range here that you can kind of work off. It's a guideline. I know some people that go lower or higher, but it's a good starting point for you to kind of experiment with. And then you've got things like reducing the surface tension of the water. So that's your kind of spreaders and stickers, all of that kind of discussion. Some of those surfactants and things, just helping to kind of spread out the droplet, help it stick to maximize surface area. Now, there's a bit of nuance here. If we're talking pesticide applications, yes, you want really good coverage. Yes, you want finer nozzles, that kind of thing. But when we're talking about nutrients or systemic pesticides for that matter, but generally nutrients, a little bit more of a coarse droplet is fine because you're looking for a systemic response. As long as you get some droplets on there, it's going to be absorbed into the plant where it can be translocated and utilized. So I think getting really fine nozzles and fine droplets, etc., really even coating is really important for particularly some of the pesticides. But for nutrients, you've got a little bit more leniency with those. But sure, a bit of spreading capacity, sticking, it does help get the nutrients in. And what that does also do, some of those spray oils, and then I think in your climate, when we come talk about humidity in a second, those oils do also help to protect the droplets. So we're going to coat those droplets and protect them so that when they land on that leaf surface, you've got an oil kind of protecting that droplet, slowing down the dehydration, slowing down the drying. And we're going to talk about that in a second. And then carbon-based inputs or chelating, we're going to dive deep into that one specifically. I won't dwell on this one because you guys are farmers and know more about all this than me. It's just all of that practical stuff in terms of use the right nozzles, not too fine that it blows off in the wind, not too coarse that it just lands on the ground. You want to hit the target. You want enough pressure there to reach the canopy, maybe penetrate a bit of that canopy. So using the right kind of forward speeds, the right pressures. You don't want to bounce those droplets off. So just those usual things that you're all already aware about. I'm not a super specialist on those. I think I hand that to you. I accept that most of those you're kind of practically speaking, you're thinking about all of that stuff already. But I think it is important just to emphasize. And then the plant itself, what plant species or what's the crop? And that's really just because some plants are a little more or less responsive to foliar sprays. Let's use an example of the brassicas. They have those very waxy leaves. When water gets on there, it really does bead. So I think on those types of plants, yes, you definitely want a nice spread of sticker to really encourage that to stick and to spread out and not bead on those leaves. So that's a kind of example that I think that's very important to include that in the formulation. But otherwise, you've got the crop stage. Generally, the rule here is that the younger leaves, newer, fresher leaves are a bit more responsive to foliar sprayers. As the leaves get a bit older, they harden off, they mature off. They still can absorb, and they still do, but they're a little less responsive. So kind of targeting it to those fresh young leaves is particularly relevant. Canopy structure, again, just making sure you want to get good coverage over that canopy, the leaf area index, get that penetrated into the canopy and over it. So kind of really coating all of those surface area of the plant. And that's, of course, relevant to then the discussion about at what point in time do we foliar spray. If you have small seedlings still and you've got a lot of bare soil exposure, might not be the best time for foliar spraying. You're going to get a lot of soil contact, which is not necessarily all wasted. Some of that can be used, but generally, as I mentioned, a lot of the losses happen from soil. So we want to try and target it onto and into the plant. So you want a bit of row closure, a bit of canopy closure there to make sure we cover up that soil. And then this is more optimum for foliar sprays. Leaf chemistry, that's that negatively charged leaf thing that we just talked about. Other things like those, I mentioned the waxes, some of the hairs, those hairy plants, that can actually be a good or a bad thing in some instances. So some of those trichomes do capture the water and funnel it down to the base, like we just talked about, but some of those hairs can actually even repel some of the water, so it can also depend a little bit there. And then other stress factors. If the plant is stressed, it's not going to be as responsive to a foliar spray. If it's under attack from pest or disease, it's got other priorities that it's dealing with, so it's not necessarily going to be the most responsive to some of those foliar sprays. That's going to kind of limit that as well. So there are some kind of plant traits, plant states that can influence overall that success. Okay, then the environment. I think this is the other big one. I think the formulation and the environment are the two big ones that I really want to emphasize. And we're going to come back to the formulation, talk more about carbon in a second. But I just want to get across these main ones here. Environment, humidity. This is key for that reason that I mentioned earlier. You want the pores in those cuticle to be open. You want the stomata to be open. And under high humid conditions is when they will be most open. That is when you're going to get more of the solution being absorbed and taken up. So high humidity, the magic number is 70% and above. If you are at 70% or above humidity, this is optimum for absorption. You guys have a lot of that around here, don't you? So I know here's the, unfortunately, now we open this conversation of the practical middle ground here. Where's the compromise? But I'm telling you, that's the optimum. You can still target. This means that time of day is key. Early mornings, late afternoons, evenings. I think in hot, dry climates like this, kind of evening times are excellent for foliar sprays. The stomata are closed at that time, but you know what? Like the humidity is a really important factor there. All the other pores are open, so I think that's still very important. And the humidity is important in terms of just slowing the drying speed of the droplet. If that droplet dries too quickly, the nutrients need time to be absorbed. It takes a couple of hours for them to be absorbed. And if that droplet dries too quickly, the nutrients can precipitate out before they've had a chance to be absorbed. So we do want good humid conditions for that absorption. So late afternoons, early mornings, evenings, this is the best. And I know practically speaking, you're not going to be able to do the whole farm at those times. So my compromise here is I would say you've got a choice here. You could choose if you've got a poor field, a crop that's really struggling, maybe you could prioritize that field for the optimum 70% for that early morning application. Choose a really poor, struggling field that needs a boost. Prioritize that one. If you've got some other fields that are doing okay, maybe they're going to get more middle of the day kind of timings. Maybe not 100% ideal, but they're looking okay. Or prioritize your unhappier fields in those more optimum times. Yeah, so at night is good too. So the stomata are closed at night, but because humidity is a bit higher, the rest of the pores and things are open. So a night is a good, in a hot, dry climate like this, I think night is also a good time. And then, if you don't mind working through the night, then that's definitely a good time. So I would just be selective about your fields. I mean, you could also, my example there of choosing a poor field for the optimum window, to prioritize in the optimum window, you could look at it another way and say, well, here's a field that's looking really good, and I want to push that. I want to push the potential of that field. So you might want to choose a good-looking field and really prioritize it in that optimum window. Now, under hot, dry climates like here, I think that where, let's be honest, you might be spraying a lot under lower humidity. This becomes, again, this important of spray oils and things, just to kind of protect some of those other carbon-based adjuvants to kind of protect those droplets and slow the drying out. I think these become very useful to reach that middle ground compromise that you might have to be spraying in other middle times of the day. And the temperature is a similar one. I've read deep into this. Kind of really, a lot of the work says a max of 88. 82. I think that is really the top end for me. I wouldn't be foliar spraying above that. I think really more ideally a max of 77, like in this kind of zone. If you can just get it a little bit when it's cooler, it is better. It's the humidity factor there. So I think the temperature obviously and humidity go close hand in hand. So trying to be as optimize that as close as you can, I think, is useful. In terms of the humidity aspects there, I don't know if you'd have this luxury here, but I know in the UK and some other parts, some of the farmers they are going a little higher on water rates. So trying to put a bit higher water in the foliar spray to try and get a little bit of kind of micro humidity, a bit of just raise the humidity in that canopy, so they'll go actually a little heavier on the water rate. So for me, I normally work at pretty much a standard of about 10 gallons an acre. I'm pretty sure that's 100 liters a hectare. So that's my kind of standard. Would that be kind of normalish for you guys around here? Would a 10 gallon an acre application total volume be ballpark-ish or a bit lower? What do you guys kind of normally around there? Getting a few nods. So maybe some of the guys just go a bit up from there. So maybe up to like 15 or something like that. Just a little bit of extra water for the humidity factor. That does dilute the spray a bit, but it does also kind of help a bit with the humidity. Okay, a little bit of wind is good for sure. Wind can really help. Just kind of coat both sides of the leaf, get that little swirl, and just kind of get some of that spray up underneath the leaf. And then this point about drought stress, drying conditions. Here I would say that foliar applications in the early stages of drought stress is very, very favorable. So when we see drought stress, when you look at plants that are wilting or curling leaves that are looking drought stress, that stress that you see is two things. One is a water stress, and two is a nutrient stress, nutrient limitation. Because when the plant takes up those nutrients, it's taking a lot up with the water. So as the soil is drying out, the nutrient availability is also dropping off. So what you are seeing is an expression of water stress and nutrient stress. And so in those earlier stages of drought stress, a foliar spray can at least address the minerals, the nutrient side of things, and at least get some of those nutrients in. And there are certain nutrients that are very good to help with that drought stress, things like potassium as an example. Silicon would be another. Some of the kelp kind of seaweed biostimulants have some good hormones in there that can also help there. So early stages of drought stress, foliars are excellent. Really, it helps the plant hold on for that next kind of, maybe if you do have some moisture coming, it'll help the plant hold on longer in the season. However, once you get further down that drought stress, once we're talking about really drought stress, the plant's really just shutting down, and it's not really even going to be responsive to a foliar spray. So I wouldn't even bother in those very late stages of drought stress. But I think early on, it's a very, very useful tool to help the plant hold on. Mm-hmm. Mm. Yeah, good point. That's a very good point. You know, I do a lot less work with maize as compared to some of the other small grains and things. But yes, you are right. You are going to have a microclimate in there. Sure, maybe those very top leaves are going to be a little more stressed from some of that direct sun and things. But as you move down into there, yes, you're right. I don't know the answer to your question. I don't know exactly what your kind of local micro humidities would be and what it's going to be like there. But yes, I can say for sure you are right. You're going to have some of the microclimate effect there, definitely. But so yes, it could well be that a foliar spray can still be effective for you, even though the overall weather, the air humidity might be much lower. It is, yeah, you make a good point there. Yes, it could still be that a foliar can be effective under those conditions because of that. Yeah, for sure. But I don't know exactly what that magic kind of number or whatever would be. But yes, you are right. Yeah, for sure. Okay, so swinging back, it's important to spend a bit of labor, a bit of extra time about these carbon additives, the extra things that go into the leaf here. I just want to touch on a few of these, and then I'm going to give you a quick example recipe and a few crop timings to round this one off. So this picture says 1,000 words. It really explains nicely why we want to include carbon inputs rather than apply the nutrient on its own, just with all of these inorganic, pure kind of nutrients. Let's add a carbon source. It acts like a sponge. It's got a lot of charged surfaces that will bind to things. And so when we mix these two together, we wrap up those inorganic nutrients. We wrap up those nutrients in a stabilized form, in a complex or a chelate. And this is why we add these carbon additives to do exactly this. Now, this is very valuable for soil applied. If you're using soil fertilizers, liquid starters, this principle holds very, very true too. you don't want your phosphorus locking up in your high pH soils, for example. You don't want that nitrate leaching away or off-gassing. So when you bind it to carbon, you stabilize the nutrient. You change its behavior. It's not going to be so reactive to lock up with all of the other soil minerals. It's already locked up. It's bound to the carbon. And it's happy being bound to the carbon where it's then stabilized. And that means then the plant can come along and get it by releasing some of those exudates, those enzymes, which some of the root exudates are enzymes that will cut open all of that carbon and liberate the nutrient, or some of those sugars that will feed the biology, and then those biology will feed on some of that carbon and also release the nutrient. So it's equally relevant for soil applied as it is for foliar. In the foliar context, what that carbon does is it makes the charge on the nutrient neutral. So we're neutralizing this cation or anion thing that we talked about earlier. We are helping then to create a molecule that will pass through the leaf more effectively. And that's what this carbon, it also helps to stabilize and etc., prevent lockups, but it is really particularly, it is helping with the absorption of that nutrient. And because it's carbon, it's now behaving just like an organic nutrient source. Now we are absorbing that carbon. So therefore, like we talked about amino acids and organic nitrogen earlier, now we are absorbing carbon. So now we are saving photosynthetic carbon. So very relevant with nitrogen inputs. If we put some carbon with the nitrogen, we are making that nitrogen behave like an organic form. So we've got embedded carbon saving photosynthetic carbon. So when I say carbon, what am I talking about? These types of things. It could be simple as molasses, sugars, humic acids, fulvic acids, amino acids, as we talked about, protein hydrolysates, which would be a mixture of those amino acids plus the peptides and the proteins. Seaweed or kelp extracts, other plant extracts, even compost extracts. We often talk about compost extracts in the context of for microbes, for biology, but there's a lot of humic substances in there, soluble dissolved carbon that comes out of a compost. So you can use compost extracts for the carbon as well, for that carbon sponge. Now these rates are just guidelines. There are very specifics on products, especially with the humics, fulvics, and aminos. They can be wildly different concentrations, so follow the label rates. I'm just trying to give you some ballpark guidelines here to also just illustrate that you can see it's not huge amounts. We're talking pretty small amounts of things that we might kind of be using, so it's not a lot. And any of these is a great carbon source. Now, I'm a fan of all of them, and I think a little bit of a diversity of them is good. Use a couple, 2 or 3, I think is great. Molasses is great for some of the soil nutrient ones, because that molasses is a great microbial food to stimulate the biology to eat your fertilizer and stabilize it in their bodies. So I think that's really good. But molasses is also a bit sticky. It's good with the foliar too, to help it kind of stick to the foliar. Now with the humic and fulvics, I'm sure you've heard of these before. I lean towards the fulvic for foliar. Fulvic is a smaller molecule, and that smaller molecule means that it passes through the leaf more effectively. So I lean towards fulvic. I think it's better for foliar. I know some people use humic, and you can use humic. It's okay. I think it's fine. But I think fulvic is my preferred choice because of the smaller size, and also because of I'm going to come back to our slide here, also because I mentioned before the pH, spray pH, and that's why I lean towards citric acid, because it's also a carbon-based acid, so it is a carbon chelator itself, plus acidifying. But because I mentioned that the spray pH needs to be more on the acidic side, 6 or below, ideally, down around 5, maximum 6, but down around 5. This is why you have to be careful with some of the humics. They don't like being acidified. They can turn to jelly. So fulvic acid, however, is fine under acid or alkaline conditions. So I lean towards fulvic because it's smaller, better for foliar, and it can be acidified, and I think acidified nutrients are important for a foliar spray. Now, amino acids, we talked about them already. They are a great source of organic nitrogen and the efficiency gains that come with that, but they are also a carbon source. They also have carbon. So amino acids are fantastic chelators in their own right. They do 2 things. So that's why I really like aminos, amino chelator. You get two for one. You have a carbon source that is great at binding to nutrients. So amino acids have carbon, so they are great at binding. They are great carbon chelators. Plus, you have some organic nitrogen embedded in there. So you've got that side of it too. So they're 2 for 1 with the amino acids, both carbon and organic nitrogen. Same with the protein hydrolysates. Kelp and seaweeds, they're also very good, great microbial foods. You have certain sugars in the seaweeds which are really good. Mannitol is a particular one. It's a really good chelator, really good getting the nutrients into the leaf. So I would choose a couple of those and use a mixture of kind of 2 or 3. And like I say, for pH, I really think citric is great because it's carbon-based. And then when we talk about foliar nitrogen, I think that urea is really good because it is carbon-based nitrogen already. Okay, so we have that carbon embedded. We get that free molecule. And urea is charge neutral. And this is why it is so effective as a foliar. It passes through the leaf so quickly because of its charge neutrality. And when you mix other things with urea, like trace elements, it's been shown really with like things like zinc and iron, lots of other nutrients. If you put those traces with urea, urea helps those things into the plant itself as well. Because urea passes through so quickly, it pulls those others as well. So there's, I'm not going to go into it today, but there's a ton of studies that show enhanced uptake of trace elements. If you spray trace elements on their own, trace elements with urea, you will get the same rate of trace elements. You will get more uptake of the traces when they are combined with urea. It's also, because of this charge neutrality, it's very, very good form of nitrogen to get into the leaf and to pull other things into the leaf with this carbon embedded. How fast are we talking? Very. This is urea here on a couple of different grasses, and this is amino acids. Your 2 fastest forms of nitrogen to be absorbed through the leaf is urea and amino acids. Aminos first, they actually are the fastest, then urea. Then ammonium, then nitrate. So nitrate is also the slowest to be absorbed through the leaf. Ammonium a bit faster, urea faster, aminos the fastest. But it's all quick. Once we're splitting hairs a bit with aminos and urea. So here we're looking at urea absorption through the leaf on a couple of 2 different grasses and on time. And just for you, those of you at the back, this first increment here is 5 hours. So you can see literally in the first 1 to 2 hours, the bulk of the job is done. Okay, and it's true on that one too. So with urea, it's very quick. It is in the first 2, 4, like 6 hours. By 6 hours, the majority of what you've applied is in the plant. And this is why urea is so favorable. Because the longer that nitrogen is sitting around on the leaf or in the soil, it's going to be prone to those losses. The quicker we get it into the plant, then we've got it. We've stabilized it. We've got it. It's not going to off gas, etc. Or it's not going to wash off. So urea is very fast. And here we have, again, amino acids. This one they had applied this one here, glutamate, which is the triangle one here. And you can see, I mean, it's literally just straight up. And these other ones kind of equally follow there. So these are the fastest forms, and this is the ones we want to lean towards for a foliar program. Urea and a combination of urea and amino acids is your top choice for foliar spray. Now, I'm not opposed to a bit of UAN. I think that, like, if you want to put a little bit of UAN in that mix, it's fine. You've got a bit of that ammonium and nitrate form. A bit of diversity of forms is okay, is good. But for foliar, I would primarily be focusing on urea and amino acids. And UAN, as I say, maybe a bit better earlier on for canopy establishment, nitrate for tillering, a bit earlier on, okay. Thereafter, I would really focus in on urea. So, therefore, kind of here's some example recipes. I've got one coming up next. Like, in terms of the structure, how do we design this? Well, we're going to start with the nitrogen sources. Do you want to use urea, UAN, ammonium sulfate is also okay, good for the pH adjustment, as we also mentioned. So any one of those, or a combination of those, combination of nitrogen sources is always good. Then I would say under maybe ideal conditions, ideal scenario, do a leaf analysis, get a tissue analysis, get a sap analysis, identify what else do I need and put those in. What other trace elements do I need? What am I actually deficient in? And put those in. That's the ideal scenario. I know in practice we don't always have a leaf analysis available, so I'm going to give you a compromise recipe next. But under ideal scenario, sure, if you know exactly what else you need, combine that with the urea and these other carbon additives. So next you want to think about your carbon sources, and I mentioned a couple there. I think fulvic and aminos, I probably would lean towards these two. I think they're very good. Molasses, as I mentioned, it's also great. Then you want to think about that pH adjustment, bring that down to a maximum of 6, but down around 5 I think is good. and then maybe any of those other stickers, wetters, etc., etc. So as a compromise, like here's a generalized recipe that's great to start with. Urea, 20 odd pounds an acre of product, 20, 25 pounds an acre of product. Kind of generally, it's going to hover around about 10 pounds of actual nitrogen. That seems to be what most people are doing, around about this 10. I encourage you to experiment from there, plus or minus. I know people who use less and say that works better. I know farmers that have gone much higher and haven't had any issues with scorch and burn. So I'm just giving you a starting middle ground. Experiment from there. And here's my kind of compromise. If you don't have a leaf analysis, etc., I would do this: a multi-trace element package. Just a standard product. I don't know your products. I don't have an opinion. Just any one that is a mixture of all of those traces. A standard kind of blend with a bit of everything: bit of moly, cobalt, bit of zinc, etc., etc. For the reason that we talked about earlier, when we looked at that metabolic pathway, we had moly on there, we had iron on there, we had nickel, we had manganese, we had zinc, we had boron. There's a lot of trace elements that are involved in using nitrogen. So as a compromise with your urea or whatever you're going to use, just put some trace elements in. Just have a little bit of everything there. At least you know that the plant's going to be able to use some of that to build those enzymes to make use of that nitrogen. Okay, then I would add a organic nitrogen source. And that's going to be your amino acids or the protein hydrolysate, fish hydrolysate, whatever it might be. So then, yep, I want some organic nitrogen there with, and then the carbon source. Okay, and again, we just talked about those. And then again, that spray pH. That's it really, and maybe other things if you know what else you need, but that's kind of where I would start. That's what I would play around with. Some of the timings, I know maybe you're not a big cereal producer region here, but on cereals, just ahead of flag leaf is a really good time on cereals. Just as that flag leaf is emerging, or just before, you're putting out that nitrogen to really grow a nice, strong, healthy flag leaf. Of course, flag leaf is where so much of the yield comes from, so kind of right onto that flag leaf as it's kind of emerging around there. Like, we've got to be flexible with the weather, etc. I know. So around there, this is a good one for yield. This helps to drive yield. If you want protein, then the other key times, very well studied, is straight after flowering, like right at the end of flowering. Some go as early as kind of mid to end of flowering or straight after. This is very good for protein. If you want that protein gain, straight after flowering is an excellent time. If you don't want protein gain, sure, I would choose another foliar a bit earlier on. If I were going around tillering, I would probably use a bit of UAN because of the nitrate, and nitrate really helps to encourage tillering. So I would maybe think about UAN there, but otherwise some other good timings: stem elongation, booting. These have been shown to be good stages for cereals as well. So something like that. You don't do any canola around here, do you? We don't pretty much do much around here, so I'll skip over that one. But kind of again towards the end of flowering is a really good time for that one. Okay. So you never know. Okay. Yeah. Yeah, I'm almost done. I've got like maybe 4 more. Yeah, cool, perfect timing. So yeah, well, end of flowering-ish is a good time, and include a bit of sulfur. Brassicas love a bit of sulfur. Now on maize, these are the times here that seem to work well, around about kind of V4, and of course I've looked into the literature on this. There's kind of a range of options here, but kind of somewhere around between V4, V5, V6 in that kind of window. Chatting to Nick last night on the drive here, he's kind of leans a bit earlier. He really thinks kind of V4 is a bit better. I know some farmers up in Canada who kind of lean a bit the same. It's convenient they combine it with a herbicide pass at that timing anyway. So I think in that kind of window, like somewhere around there, V4, V5, maybe V6, seems to be good. Another one is somewhere around V10 or V12. I've seen kind of mixed kind of results around there. Again, chatting to Nick last night, he really thinks that a bit earlier is better. So he was kind of more of a V10 kind of guy, but kind of somewhere around there. And then I think really kind of straight after flowering, like that's also a really good one for kind of late season quality, etc. as well. So that's kind of 3 windows. I mean, the world's your oyster. You can do as many as you want or as many as you can, but I've just kind of cherry picked some of the key timings. And then on even pulses, on a range of different pulses, this has been shown also on your soys, but I mean lots of others. Obviously, you don't want to be pumping too much nitrogen into them. You don't need to, but late season applications can really help with protein. if you're looking for that protein gain. So again, kind of really straight after flowering, around pod set. There's some good work that's shown around that timing is kind of really good, and really it's more about that protein bump. And I wouldn't be putting on huge amounts, maybe 5 pounds of actual N, something like that, 10 pounds of urea. You could play around with that, but just I wouldn't be putting huge amounts on. I don't think you need to. You could play around with that, but obviously I don't think you're going to need quite so much. And you wouldn't want to put it on earlier because of you don't want to maybe suppress too much of the end fixation, etc. Although that said, I've seen a couple of really interesting studies that show that actually a foliar application of nitrogen is much less suppressive, and in some instances even not suppressive, of end fixation. And that's really because the signal or the trigger to stop end fixation, or for the plant to stop feeding the rhizobium, is primarily nitrate in the soil. So when nitrate level builds up in the soil, that nitrate is a common what's called a signaling molecule. So it's not only is it a useful nutrient, it also is involved in sending signals through the metabolic reading of the plant. And so when nitrate builds up around the soil, that is the signal that tells the plant, okay, there's enough nitrogen around the soil that I can then continue to scavenge for the rest of the season. I don't need to feed the rhizobium anymore. So this is the beauty of the foliar nitrogen, is that you don't short circuit that, because if you can keep nitrate from building up in the soil, you can still top up through the foliar without compromising the end fixation. But I still wouldn't go too early. I still try and let that go as best you can and think more about the late season applications. Okay, lastly, shameless plug. Last year, about this time last year, I ran a detailed online course called Foliar Nitrogen, all about this. You have today received the key kind of highlights and summary of that, but I went a lot deeper into this. If anyone's interested, it was 10 lectures, over 10 hours of content of what you just heard the summary of. I go really, really deep, share a lot of different studies about all of this. We recorded everything. It's all up online on my website. If anyone's interested, if you want to kind of hear more about that, that is available to purchase, and it's really not expensive, like $200 US, kind of in that ballpark. You get permanent access to that to watch as many times as you want. So that's available there. If anyone's interested, you'll find it on my website, integratedsoils.com, or just type in Joel Williams and soil, and you'll find me. Okay, in closing, some of the key highlights from this morning's lecture: combine inorganic with organic. That's one of your key messages. Always include a carbon source. I think aminos are good there because they're ticking 2 boxes. Drop that pH down around 5 if you can. I know some farmers will go even lower than that, and I think that's okay, but I think aiming around 5 is good. Use your plant health assessments, tissue tests to maybe determine what else you need. Maybe use that multi-trace element blend as a compromise, as I mentioned, and think about that FACE formula: formulation, application, crop, environment, with particular focus on tweaking your formulation and trying to optimize the timings of those. I think are 2 best kind of bang for bucks there. So, okay, good timing for lunch, I guess. Thank you.
Before we break for lunch, I have just a couple quick things. They won't take long. So I am part of the Kansas Soil Health Alliance. I'm the coordinator of it. We've been around for a few years. Many of you have hopefully heard of us or found information about today's workshop on our website. A couple of things that I